Very much like you for putting this together, and I've really enjoyed the talk so far. I thought that I would speak about the role of herbaria. So I'm the director of the herbarium. I have been for some number of years. And I want to discuss its role in botanical discovery. And so my talk is going to be about sort of the role of the herbaria in Amazon biotic exploration. Let's see. Okay, so first I'm going to talk a little bit, give you sort of the big picture view about tropical forest tree diversity. That's my primary focus, is tropical trees. And then I'm going to talk to you about how I've used the Brazil nut family, Lecythidaceae, as a model system to sort of explore some ideas about the diversification history of the forests. And then I will use this to sort of bring in the herbaria. Where does an herbarium fit into this kind of research? So this is what really has been my driving interest even since high school. This is what I wrote my college essay about, was the latitudinal gradient in tree species diversity. And it's really well represented in this slide, where we have three big forest biomes represented. We have our boreal forest in blue, we have the temperate forest in red, and then we have the tropical forest represented in green. And you can look, each number associated with each region is the number of tree species that are estimated for that region. So you can see, just looking at the Americas, we have 61 tree species in the boreal region, 115 western, 300 in eastern North America, and then 22,500 tree species estimated for the neotropics. So here you can see 161, 1,600, 43,000 tropical tree species. And when you take a really close look at, really intensive look at a single rainforest in a very small area, like 1 hectare, you can find 300 tree species. So take a look. Oops. We've got 300 tree species in eastern North America and 300 species in a single hectare of forest in the middle of the Amazon. So this is a big question, like how do we get so many species? I've been interested in the historical approaches to addressing that question, and I tend to think back over long periods of time. So we can think about the rainforest as it exists now being established about 65 million years ago after the KT impact of the asteroid. So we've got 65 million years of tropical forest. Tropical forests have not always been just tropical. They've, at various times as the Earth has been warmer, tropical forests have expanded all the way up to Michigan. So you can see in this Paleogene era, 65 to, especially 65 to 55 million years ago, we actually had a tropical forest type of vegetation in Michigan. So very broad. It was like the predominant forest biome around the world. And we have the Neogene period, 24 million years ago to 7 million years ago. Tropical forests still very extensive. So we look now, and now, even though tropical forests are expansive, they don't represent as much as they did previously. My lab's focus is in the Amazon region, and we're very interested in understanding how the uplift of the Andes helped to create the Amazon basin and how that led to plant diversification. So at some point before the Andes uplifted, there was no Amazon drainage system as we know it. It took the Andes rising up over the past 65 million years to change the course of the various drainage basins and push the Amazon into the direction toward the Atlantic Ocean. And as that uplift occurred, it created many geological novel landscapes for plant diversification, and that's what we're interested in. And so a question we ask, what are the major zones or geographic areas that are associated with plant diversification? And when did these major evolutionary events occur? So I'm not like Stephen Smith. I can't put up a phylogenetic tree with 3 million species, but I can provide this cartoon of a couple species. And this is the model. This is kind of my framework for thinking about diversification. You can have a museum model where we have 65 million years to the present, where you have a gradual diversification and accumulation of species diversity. On the other hand, you could have a model, another simple extreme, where you have very long branches where there's potentially been lots of extinction, but most of the modern diversity is very recent and sort of a radiation, a burst of speciation. These could be called a museum model and a cradle model. And so these are just two kinds of diversification scenario. In this one, you might expect that these forests have been around 65 million years. There's been ample time to accumulate diversity. Here you might say that even though there's been ample time to accumulate diversity, the most important events were the recent uplift of the Andes, emergence of the Isthmus of Panama, Pleistocene climate change. So in order to address that, we really need to have model systems of tropical trees where we can work out their phylogenetic relationships, work out the geographic ranges of all the species, and really know each species that is part of a given clade. So a few years ago, I decided to focus on the Brazil nut family, Lecythidaceae. This is a Brazil nut tree. These are phenomenal, majestic trees in the Amazon basin. This is what the fruit capsule looks like. It's woody. You have to, it's terrible for seed dispersal. I don't even know how these get dispersed in the wild. But people will gather these, cut them with a machete, and these are the Brazil nuts that you get in a nut container in your house. There are about 10 genera in the Neotropical Lecythidaceae, 232 species. It contains the most abundant species genera of trees in the Amazon basin, which is Eschweileria, and it's about the third most abundant family in the Amazon basin in terms of tree numbers. I love natural history. These are one of the many beautiful flowers, complicated flowers in the Lecythidaceae. They're either pollinated by bees or bats. This is Brazil nut collection. This is a tree called Cariniana legalis, a very valuable timber tree that's protected from the Brazilian Atlantic forest. And there's this cannonball tree, a very odd-shaped tree with flowers, just a few representatives. So postdoc Oscar Vargas has been getting together all the genetic data available for the taxa, getting all the geographic information, and we put this together in a phylogenetic reconstruction. And Oscar is co-supervised by Stephen Smith, which helps a lot with the large data analysis. And so when we divide the neotropics into these zones and then associate each species with a particular zone, we can reconstruct the ancestral area of origin. And what this preliminary analysis indicates that there are a couple of centers of diversification of the Lecythidaceae. One is this Guiana Shield region right here in green. Another is this northern Andean region in Central America, often associated with cloud forests. There's another diversification. So that's just based on our preliminary data, but that's how we're starting to sort of reconstruct this diversification history. How much time do I have, by the way? Okay, so in order to work out the geographic ranges of these species, we have to identify them throughout the Amazon basin, and it's very hard to identify all these species down to the species level because you need flowers. And you can't get flowers easily. Sometimes these trees will flower once every 5 years. And then you have to climb to get the flowers. So there's really imprecise taxonomy, and that limits our understanding of the species range. And it also means that we're actually discovering a lot of new species. There are new species being described every year. And so my lab we really wanted to get in and actually learn these species in the field. And so I found an inventory plot that was established in 1988 of Lecythidaceae, all the Lecythidaceae in 100 hectares of rainforest. And all the species were identified by the specialist Scott Moray from New York Botanical Garden. We decided to revisit these, get genetic information, learn these species in the field and their field characters, and get botanical vouchers for the herbarium. So all in all, there are about 8,000 individual trees and 40 species, good percentage of the whole family. And it was located near the city of Manaus, the Biological Dynamics of Forest Fragments project. So this is where I spent a good part of my summer. This is also just happens to be where I did my PhD research in the 1990s. So it hasn't changed one bit. If anything, it's gotten worse over time. We sleep in hammocks. We bathe in this cesspool here. It used to be nicer. And we do our botanical collections and bring them back to the camp. So when you're in the field, the external bark, we can use the external bark to identify a lot of trees in Michigan. But here, it's actually the inner bark. You have to use your knife and actually scrape into it. And the inner bark of the Lecythidaceae contains a whole variety of colors that are consistent within a species. That's one feature that's used along with a whole range of other characters that you can identify in the field that you don't find in the field guides. So the woodsmen who work in these areas know these species in this way by these field characters. When we do find trees that are flowering, we make these collections. We bring them back. We collected from 3 species that haven't yet been described by science. We've got the DNA, brought them back. So here's how you study Lecythidaceae in Michigan. You go to the herbarium. This is our herbarium. This is one of my graduate students, Tamara Milton. This is what Lecythidaceae fruit case looks like. And so this is where we do a lot of the sorting out and understanding more about the biology of the group. The University of Michigan, people complain it's not on Central Campus, but it's a lot closer than the Amazon basin, less than 5 miles away. Beautiful building, and 1.75 million plant specimens, plant and fungal specimens. This is a specimen that I collected in 1992. And you can see what has happened. This resides at the New York Botanical Garden. This shows what's happened to that specimen over time. So I collected it in 92, and I actually identified this tree. A duplicate specimen was sent to Michigan, so I go right into our herbarium and get DNA from it. At one point, it was barcoded, the specimen, digitized, imaged. Somebody came and studied the leaf venation as part of a taxonomic revision, included a photograph of that. Then they later changed the name for the genus, so somebody had to correct it. It's now Allanthoma instead of Coriniana decrantha. All that information is on this specimen, which is now, this is an image that I just pulled off the internet. So this is a big project that we're doing right now with our herbarium. We want to make all of this collections and all of this collection data available to the public, available to researchers. And so we no longer have to sort of go into the forest or visit individual herbarium museums. People can sort of download it, pull down trait data, get geographic distribution data, verify species. And so we've been working a lot on this. We've had 7 NSF grants, and we've so far imaged or databased about 600,000 of our specimens or more out of the 1.75 million, making good progress. And just on a final note, there was a paper that came out in 2010, Herbaria are major frontiers for species discovery. What they say is a lot of specimens that are collected in these remote locations, they'll sit in a museum or an herbarium for up to 50 years before a specialist will come and make sense of that collection and say that, I've been studying this group for decades. I've never seen a specimen quite like this. Then that's where that specimen can be visited, maybe described as a new species. So this study, they estimated that about 1/4 of the new species that were described in the previous years came from specimens that were collected over 50 years ago. So a long-term value. They estimated there are 70,000 plant species remaining to be described, but about 35,000 of these are already in herbaria, just waiting to be visited and studied. Conclusion, invest resources into herbarium discovery as well as field-based expeditions. Thank you.
So, Joe, how do you set up a heat talk? We can take a 1 or 2 questions for Chris.
Is the data input method standardized across different institutions?
Yes, there's sort of a Darwin core data standard that's been adopted not just by the herbaria but also the zoology collections, so we get a certain kind of data. And some of the key data is like geographic locality, collector.
I have some. It makes me think that before, say, to collect the herbaria specimens, we have to travel to all over the world to go to the field sites. But now with these specimens digitized, of course, it can be accessed by people all over the world. But would that discourage the people from visiting herbaria all over the world to see the specimens?
It will in some cases, but there are actually, I think the benefits outweigh the costs. I think many, many more people are studying the specimens, even though they're doing this virtually. But it does decrease the traffic, but it makes it more accessible to people in other countries as well.
Thank you.